Okay, so what I really wanted to do was use what forced us into lockdown in the first place as a little bit of a tool to go back, especially for the advanced tires, over really everything we've covered in advanced tire. But it seemed like a really good opportunity to do this with the chemistry uh, pupils as well. The idea here will be that I'm just going to talk through really from the very basics right the way up to how our cells are infected by this particular virus, and we're going to go into some of the things that have been spoken about in the scientific literature. None of that is going to be too heavy on here, but I will, especially for those who are more interested, all the references will be in the description box. So what we're going to start with is we have genetic information. So for those of you who have maybe forgotten or haven't covered this, genetic information is tied up in either a DNA molecule or an RNA molecule. And all that molecule does is, well, all it does. In terms of what we are thinking about, all we want to focus on is that it will give you the order that the amino acids come in. So there's about 20 to 26 amino acids. I think the human body uses about 20 different amino acids, and they can be put in a very specific order to give you a very specific protein at the end. So proteins are made up of these things called amino acids, and this is one of our amino acids here. Now, what we've got is an amine group and an acid group. This is actually the type of acid section that you would find in something like vinegar. So this is a carboxyl functional group, and carboxylic acids all have this functional group. That H may pop off in a dynamic equilibrium to create an acidic environment, but that's not really what we want to focus on going forward. What we'll focus on is how these two are connected together, and then what actually happens with this string of amino acids that we've all connected together. So we'll just say that there's 20 amino acids. In fact, it might even be a good idea to show you these amino acids. So, these are our 20 common amino acids. That's fine. And the R group is the thing that's different between each one. Each one's got this amine group, and we've also got the acid group there as well. But in the middle of this molecule, we might have an R group jutting out, and that's what's different in each one of these, and it gives them different properties. Importantly, the acids have a little negative charge there, if you can see. So aspartic acid, glutamic acid have negative charges. Arginine has a positive charge, and that's basic, and that's going to be really important for the SARS-CoV-2 spike protein, which eventually we'll get onto. So that basic arginine with a positive charge, really, really important. So those are the R groups for the different amino acids, and all you really need to know about them is some are positive, some are negative, some are polar, which means they have a slight charge, and some are non-polar, meaning they don't have any charge at all. Now, when we link these together, that linkage will happen at the ribosome. If you don't know what the ribosome is, it doesn't really matter. These two are linked together by the exclusion of this hydrogen here and the OH here to produce water. Because water is being kicked out, it's called a condensation reaction. And we will form a peptide bond. Now, it's important to note that our nitrogen has a valency of 3 and it's got 3 bonds, and they are all what we would term covalent bonds, so there is a share of electrons between the nitrogen and the carbon, the nitrogen and the hydrogen, the nitrogen and that hydrogen there. So the peptide bond is a covalent bond. It's just a really important type of covalent bond, so we give it a special name. Now, from the genetic information, hopefully this works, that will tell me what amino acid I'm using, and it'll put it into a very specific order. So the order my genetic structure is putting these amino acids into with their specific little R groups is pink, green, pink, red, blue, green. And that order we call the primary structure. So that is just the order in which these amino acids are placed. You may have a protein that is 200 aspartic acids long, and it's just all aspartic acids, and then on the end you drop an isoleucine, and so that would be the order. You can do lots and lots of you can order these however you like, and the order will give you that primary structure, and that's really important in creating the final protein. So that there is really the first thing that you need to know. The R groups here, say that was positive here, and that was negative here, and maybe that one was non-polar. There would be a force of attraction between that positive and negative, and it would contort towards each other. And that would give you a very specific conformation or shape between that section. That's really what we'll move on to with the secondary structure. So I'm kind of hoping... I'll need to... Okay. So once we've got our primary structure, we start to get this contortion between these amino acids dependent on the specific R group that we've got. So depending on what R group we've got, we'll get a specific movement, and those create specific motifs. So the motifs are really either an alpha helix, an anti-parallel beta sheet, which is where the order of amino acids, if we've got, I need to do this again, if we've got amino acid 1 here, that would be 5, if that is 7, that might be 10, and the stream in terms of order is going from left to right and then back from right to left, and it's anti-parallel. A parallel beta sheet, the number or the order of the amino acid is all running in the same direction, and so you're not going to get that quick turn. You're going to have the sequence go off and do other stuff and then come back around, and that's going to give you a parallel beta sheet. So these are what we call secondary structures, and these are really the big obvious characteristics that you see forming from the polypeptide chain, and really the basis of most of our proteins that we get will be a bunch of alpha helixes, a bunch of beta pleated sheets, all contorting into different shapes. For the advanced hires, just remember any bits that aren't alpha helixes or beta pleated sheets are turns. So the bits connecting these major motifs you can think of as turns. So this is our primary structure up here, and that's just the order of amino acids. This is our different types of secondary structure that we get, and that's really it for that bit. So, tertiary structure. Once we've got these alpha helixes and beta pleated sheets, they will form up into a three-dimensional shape. You can kind of imagine this as just like a bit of plasticine, like it's finally taken up a three-dimensional shape. Some polypeptides are ready to go and do their job as soon as they take up that three-dimensional shape, so they are known as proteins. If it's performing a job, you can say that polypeptide is a protein. However, not all polypeptides are ready to go once they've been translated. What is going to happen in most of those polypeptide chains is they're going to have some modification done to them after translation. And so specifically what we have for the spike protein in SARS-CoV-2 is we have a homotrimer, which is basically we get the production of 3 polypeptide chains that are the same. Then, as a post-translational modification, those 3 polypeptide chains are bonded together, and then specifically once again in SARS-CoV-2 spike protein we get our sugar molecules being attached. The oligo just means group of saccharides, which are just sugar-based molecules, so you can just think of them as sugars. So what we've got is a bunch of amino acids that have all contorted in shape due to their R groups. They'll take up alpha helixes and parallel and anti-parallel beta pleated sheets with a bunch of turns. Then we get some post-translational modification where in this case we get 3 polypeptides pulled together, and then we get some of these oligosaccharides attached, and we get our final quaternary structure of our spike protein. And all of those modifications that we do after the initial building of the polypeptide chain, those are post-translational modifications. And it's really cool and important to know that the coronavirus, no virus is able to produce its own proteins. These proteins are created by your cells. The virus lacks all the machinery required to produce these proteins, and so they need to get into a cell, and the DNA or the RNA of the virus will then start producing these proteins inside your cells. So it's important to know that actually your cells are complicit in the building of these spike proteins on the virus membrane. So what I want to do is first of all, I realised that for any of my National 5 group, this might have seemed a bit much. It is advanced higher. All you really need to know if you are some of the National 5s or some of the higher chemistries, that we've got really what I want you to be thinking about is we've got a bunch of atoms that are all flung together, and they take up a very specific shape based on the genetic information. And that specific shape that we're interested in is the SARS-CoV-2 spike protein. It's sometimes called SARS-CoV-2S for short. These are really the most important proteins for the virus. It's how they get into cells. It's why we call it a coronavirus. So we'll probably take a little look at the electron microscopy of a SARS or a coronavirus, and you can see those spike proteins, and that's why it's called a coronavirus. So we're gonna now look at this guy here. Do it in green. Pymol always reminds me of green. We are going to take a look at this one in Pymol, which is a program designed to show us what this protein looks like, and we'll talk a little bit about where we find different things, and then we'll take another look at where this protein is, and how it functions between the viral particle and your cell. So, this is not our spike protein. This is actually just a single polypeptide subunit of that spike protein. And what I want to do is talk about this first, and then we will add on our other polypeptides to see what the full thing looks like. So, for those of you who haven't seen this program before, it's Pymol. The advanced hires have. There is no need to be able to use this, but it is really nice for trying to visualize what's actually going on. If we look up here in this section, we have got arrows that are going in the opposite direction from one another, so that would be an anti-parallel beta sheet. Now down and round here. Here we go. So here the arrows are still going in opposite directions, so that's an anti-parallel sheet as well. They're all anti-parallel sheets actually. And this section here is chock full of alpha helixes. Now this is going to be really important. These alpha helix sections are going to be really important for integrating our spike protein into the membrane of the virus. So basically, to cut a long story short, the inside of the membrane is non-polar, which means you need a non-polar thing to be stuck in it, and these alpha helixes, at least the outer side of them, is pretty non-polar. The inside might be polar, but that won't affect anything. They display as being non-polar, and they will sit inside the bilipid layer of the membrane. So what I want to do is I want to add in our will it be wires or cartoon? I think it'll be cartoon. And subunit 3 show as cartoon. Right, so what I've done there is I've added another 2 polypeptide chains, and they are now bonded together because of some post-translational modification, and we've got these 3 polypeptide chains in a quaternary structure. The sugar molecules in this particular one I don't believe are shown, but it doesn't really matter for the purposes of it. This bit here up at the top, so if I orientate it like that, this bit up here where you can see the mouse cursor, that is the receptor binding domain. That is the bit of this protein that's going to come into contact with another protein on your cell, specifically called the ACE2 protein. And the better a virus has mutated to infect humans, the better the receptor binding domain will be at attaching to a human protein. We'll take a little bit of a look at that later on. You can see that this section of alpha helixes down here, they will be embedded into the membrane of the virus. They're also really important for allowing the virus membrane to fuse with the membrane of the human cell, and that allows the viral DNA to then get into the cell. So this bit here is really important for attaching to a human protein, and the bit at the bottom here, that's really important for allowing the fusion of the two membrane structures to come together. So this is not the SARS-CoV-2 protein. What I want to do is really show you where we can find that, display that, and then have a little go at seeing if we can find something interesting using one of the papers that have been released for SARS-CoV-2. So, what I'd like to do is go to... Oh, it was the Queen's birthday on the 13th. That's Wikipedia. Where have I put it? So this is our SARS-CoV-2 open state. I think that means that it might be in the state where it's bound, but I'm not sure. I don't want to say things that I'm not 100% sure on. So this is the code that I'm going to use for producing this structure in PyMol. So PyMol. See if this opens. I think it's opening. Fab. So I want PyMol to go and fetch this for me, and so I just type in the code and it'll bring up the protein here. Looks to me like we do actually have... Oh, make it a bit bigger. We do these little things here. They look like our sugar molecules potentially. Oh, what I want to do is show this here. So yeah, those look suspiciously like sugar molecules. Well, maybe not. No, that's a residue. Okay, ignore me. A little bit odd. I think those might be residues from another protein. We're not going to worry about that too much. It's not really what I want to show you. So, this still has all three subunits, but I haven't color coordinated this because I've just brought it up. When you bring a protein up in PyMol, you can then start doing interesting things. You can measure the distance. You can see what type of bonds are present because of the distance. Lots and lots of different things. But what I want to do is look at a paper and then find the bit on the protein that the paper's actually talking about. So one of the really interesting things about the SARS-CoV-2 is that there's an enzyme in the human body that can chop a little bit of this off and make it really effective at infecting human cells. So as the SARS-CoV-2 protein is unclipped, it's not very good at getting into cells. But once there's been this chopping by this enzyme called furin, then it can really easily get into our lung cells. And you've got that enzyme just in your mucosal membranes, so that enzyme can act on the little site that's called the furin site on the spike protein, and it'll chop a little bit of it. What I want to do is actually find that site within this program. Now that's going to take a little bit of... This took me a little bit of time to get right, and I'm hoping I can film it in one go. So what I want to do is use a scientific paper to just find a specific bit on this protein that they were speaking about. And so what I'm going to do is I am going to go to the paper that I actually want to talk about. And it is this multi-basic cleavage site. Basically, if I've still got it, arginine's basic, what they mean is, by multi-basic, is we're going to find or polybasic, we're going to find a cluster of these arginines in part of that big protein that we've got here, and that multi-basic site is going to be chopped and changed by an enzyme that we find in our body, and that chopping and changing is what makes it really good at infecting our cells. So this is something that the advanced hires will have just been introduced to, really, is scientific papers, and they can look really scary and intimidating. Proteases, for example, if you don't know what that means, you would have to look it up. There are lots and lots and lots of words that I don't know in any given paper that I try to read, and I take a long time just Googling what each thing means and slowly building up a picture of what they're actually talking about. So they've used one of these protein modelling systems to produce this image here. And basically this is our alpha helix blob. In fact, what I might do is, if you take a wee look at that, what I can do is I can change this into that by giving it a surface. It's loading. Let's hope. So I can turn that into that by giving it a surface. And it's not color coordinated like the one in the paper. They have selected different areas to color coordinate. So the areas that they have selected to color coordinate is the receptor binding domain. That bit in purple there, that bit is going to attach to your human protein. That's what's going to anchor it on to the cell. The S1 region is all of this blue and purple bit, and that is all responsible for the interaction with the human protein part, and that's called an ectodomain because it sits outside the viral particle. This bottom bit here, that's called the S2 domain. That's the bit that we can see in... Oh well, this one's still good to go. That's that alpha helixy bit down here. So that's that alpha helix bit there. That is all... Oh, find it again. That's all this bit here. That's really important for anchoring the spike protein into the membrane of the virus, and it's also really important for fusing the viral membrane with the lung cell membrane, but we will get onto that in a little minute. So if I scroll down, blah 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 blah, all this stuff, this looks scary. It's not scary, it's really quite straightforward, and what we've got is all these letters represent amino acids. So as you can see, each of these amino acids has a single letter code. It's also got a 3-letter code. This is the DNA code that will produce it, but you don't need to worry about that. So all these letters are just the single letter code for the different amino acids. So what I want to do is I want to find this region here, because this is the region that's different about SARS-CoV-2, and it's different because it's multi-basic, and we'll get on a little bit to what that means in terms of whether or not this evolved or it came out of a lab, which would be fascinating. But we'll have a little discussion about that and how to formulate a question around that. So what we've got is our code here, and I want to be able to find this bit of my protein. So right, just before we do that, to make it obvious what we're looking for, this code here is shown as those red splotches. So there's different ones, and we are picking out hopefully one of these cleavage sites. So the S12 cleavage site or the S2 prime cleavage site, we'll take a look at. Now, I could scan through, I want to deselect everything. I could scan through this entire stream of amino acids looking for the code that's in that paper. So what was our code in the paper? GICASYQTQTNS. I could look for that manually, or I could steal someone's hard work and I could add in a search program here. So what I'm going to do is I'm going to go into File and Run Script. Now this is what's really cool about a lot of these programs. They have been developed initially and then other people have added things onto them because of what they wanted to do. Now I'm lazy, I don't want to look through all that code to find that specific sequence. So if I hit Run Script, what I can do is basically add on a Google search system. So I'm sure I put it in Programs, PyMol. Now there's nothing here because I've not all runnable, but this is what I want. So this is basically a Google search system, and by running that script I can now search for this thing. So I'm going to find sequence, and the sequence that we saw from the paper, which I've written down on a piece of paper so I can actually remember it. I don't need the whole thing because the likelihood of 6 or 7 amino acids coming up on a consistent basis is pretty low unless it's a repeated sequence. So what I'm going to do is I'm going to search for the SYQTQTNS. Now what this is going to search for is letters that match that in the protein amino acid code. So what I need to do is put in this protein specifically has the code 6VYB, and I need to make sure that my search is run on that, and hopefully, look at that, we get 3 of them. And are they where they should be? Well, they look pretty close to where they should be. What I'll do is, where I've found these sections, I want to rename that section, and I'm going to rename that cleavage site, and I'm going to colour it red, will stick out. So these bits here are where we are. I'm hoping you can see that. I can zoom in, but not too far. So these little red bits here, those are the bits of this protein that an enzyme is going to chop, and it's going to chop those bits in a specific way that allows this protein to bind with human cells in a much, much stronger way. So next little bit is actually back to this lot, and what I want to do is take a look at this. Right, so what we've just done is everything that you've just seen in that program PyMole is this spike protein here. So these spike proteins are all the same, and they're embedded in the membrane. And this bit here is that alpha helix there. And that's embedded in the membrane. This is our receptor binding domain, and that's what needs to bind to a human cell. So on our human cells, we have these proteins called ACE2. And I've simplified this up, but basically the charge, the polarity, the shape of that spike protein along this section will be complementary to the ACE protein. So this is our receptor binding domain, and it's going to try and attach onto there. Now, it can't do that very well unless our enzyme, furin, chops a little bit off the spike protein, and that allows it to work. So that enzyme is your enzyme, and it's chopping a little bit off the spike protein to allow it to bind to that ACE2 protein, and then it's going to move through the cell membrane. And the RNA that's encoded here, that is going to be able to get through, and then it's going to produce lots and lots of viruses, and those viruses are going to leave the cell and go and infect other cells. And the more they do that, the more damage they cause, and that's what causes pathology or disease. The root of disease, I think, is a pathology. So disease is you not functioning perfectly. So that there is the spike protein and the SARS-CoV-2, specifically spike protein, and why it's really infectious is because of this little furin site that we've got. Now, the very last thing that I want to talk about is how to formulate a question. So I'm going to close that, and I'm going to bring up our very last picture. So there are 7 coronaviruses that cause disease in humans. There's SARS-CoV, MERS-CoV, and SARS-CoV-2, which is the one that caused us to go into lockdown. The ones circled in red are very closely related, and they cause severe disease. The ones in blue, they are closely related to each other, but not closely related to the ones in red. So these ones here are closely related to each other, but not very closely related to these guys. And the polybasic cleavage site, that is what we spoke about here. So these little red tagged bits, if they're polybasic, they are really easy for that furin to clip, and that makes them highly infectious. So HKU1 and OC43 have polybasic cleavage sites, and SARS-CoV-2 has a polybasic cleavage site. Now, the interesting thing is that the SARS-CoV and the MERS-CoV that are closely related to the SARS-CoV-2 don't have those polybasic cleavage sites. So the question is, and this is something that's covered in biology and has less to do with the chemistry stuff, but it's quite interesting, is the SARS-CoV-2 convergently evolving to be like the HKU1 and the OC43? So has evolution allowed this guy to look like these guys, even though they're not that closely related in comparison to these two here? Or did a scientist, for whatever reason, take information out of this one and put it in this one? So that is the question as to whether or not it came out of a lab. Did that polybasic cleavage site come from convergent evolution, which does happen and it happens a lot, or did that polybasic cleavage site get taken out by a human from one of these two and put into the SARS-CoV-2? The answer to that question is quite far from being answered. If we were to discover a SARS-CoV-2 viral particle that showed some intermediate steps in its evolution, that would be really good. However, that is like finding a needle in a haystack. If the needle is the size of a virus and the haystack is the size of Asia, and it's back in time. So it's going to be really hard to get a step-by-step convergent evolution approach to that. The other thing is that if all the work on coronaviruses have been published, then it's almost certainly convergent evolution, because the science to do the orange line has not been published anywhere, really. It's possible to do it, but it's not been, there's nothing in the literature that would lead someone to believe that that's what happened. So either, I'll do another line, in fact I'll do green for natural way. This one could have convergently evolved to look like either of these two, or unnaturally information was taken from these ones and put into the SARS-CoV-2. Now, I am way, way far from an expert on any of this stuff, but hopefully this got you a little bit into why it's really important that we do the science that we do, why we learn about the science that we do. In terms of your understanding, this starts off at National 5. So at National 5, we really speak about this stuff here, and we speak about how these bonds happen and how they're covalent, and then at higher we move on to this little step, and then advanced higher we move on to the next steps that we saw. That all accumulates in you building up an understanding of what's happening, and it also eventually will allow you to take some information from these scientific papers that we've got. Guys, that is the little bit that I wanted to do to make you guys see how this stuff that you do in school will eventually connect with the big things that are happening in the world. And the more you pay attention now, the harder you work, the more you go over things, because I forget stuff and I need to go over it and over it again, the better you'll be able to pick these things up when you get out into the wider world and you've got to figure some stuff out for yourselves. This is not in any way, shape or form a perfect presentation of what's happening. I'm sure I've made some mistakes along the way, but hopefully it was interesting, and yes, let me know on Teams if you liked it and if you've got any questions. I realise that I will definitely have lost people along the way, but hopefully it had some interesting little points to it.